Our next topic that we're going to take a look at is eukaryotic cells, which is similar to prokaryotic cells. Eukaryotic cells are a little bit more advanced, have a little bit more structure. They have organelles, obviously, in comparison to prokaryotic cells. So we're going to take a look at those for IB 2.3. Um, some kind of basic background and some characteristics of eukaryotic cells. One of the primary things that distinguishes a eukaryotic cell from a prokaryotic cell is that the DNA is contained within within a membrane-bound nucleus. And so in our cell image here, here's our nucleus, and the DNA is actually contained within this nuclear region. The nucleus has a nuclear envelope or a nuclear membrane, which kind of acts like a plasma membrane in terms of letting things in and out of the nucleus. Eukaryotic cells have membrane-bound organelles. They include both plant and animal cells. We'll look at some differences between the two of those, and they're actually usually quite a bit larger than prokaryotes. Eukaryotic cells are usually about 10 to 100 microns, whereas prokaryotes are usually not much larger than 10 to 15 microns. So some pretty big size differences between the two as well. Let's take a look at some of the structures that you're required to know for eukaryotic cells. The first is obviously the nucleus, as we just discussed. This is the region where the DNA is kept, and so here is our nucleus, this large purple sphere in our image, and here are some pores within the membrane that allow DNA to enter, excuse me, not DNA, RNA to enter and leave the nucleus, and this is basically where the DNA is kept within the cell. The primary purpose of a nucleus is to help. If the DNA were to be damaged, obviously the cell wouldn't be able to carry out its functions that it's required to do in order to ensure the survival of the organism or that organ or whatever that cell is supposed to do. Our next region is the nucleolus, and this is a specific region within the nucleus, so it's this darker portion, purple here. And we'll take a look at what the nucleolus does. It's a smaller portion of the nucleus. The rough ER is this series of primarily transport molecules here, structures around the nucleus. The thing that separates the rough ER, there's our rough ER, from our smooth ER is that the rough ER has ribosomes attached to the outside. And we'll look at the specific functions of that as well in just a minute. The Golgi apparatus, located right here, usually associated with vesicles, either coming to the Golgi apparatus or leaving the Golgi apparatus. Ribosomes, here are some ribosomes that are free-floating in the cytoplasm like prokaryotes, but unlike prokaryotes, we have ribosomes that are found on the endoplasmic reticulum, right along here. Mitochondria, kind of the powerhouse of the cell, or one of the organelles that produces energy in cells, in eukaryotes. And the lysosome, it's kind of like the garbage disposal, or what helps to clean up the cell. So let's take a look at these organelles a little bit more closely and their specific functions. Here's another diagram that has some nice labels of different organelles within a eukaryotic cell. And the first one that we want to look at is the lysosome. Let's see, where is it at in this one? Right here, lysosome. And really what this does is it contains enzymes that help to digest macromolecules. And so if the cell has foreign particles or if it has waste that it needs to get rid of, it's sent to the lysosome. And the lysosome kind of acts as like a garbage disposal. It helps to break down whatever those macromolecules are so that the cell can get rid of them or to remove them so that it doesn't harm the cell. Waste is then removed by vesicles, as we'll talk about in some different processes of endo and exocytosis. The nucleus, as we mentioned, holds the cell's DNA, and by holding the cell's DNA, it helps to maintain the cell functions because the DNA is what ultimately is used to produce proteins, as we'll talk about, and those proteins kind of control and carry out the functions of the cell. The nucleolus, right here in the center of the nucleus, it's a portion of the nucleus, and it's responsible for producing ribosomes. Ribosomes are what produce proteins, as we just discussed, and so obviously the nucleus then is a very important portion of the cell. The rough ER, or abbreviated little r, capital ER, it secretes proteins because there are ribosomes attached to it. So here's our rough ER right here. And it grows by adding membrane proteins. So it can actually grow and expand. You're not required by IB to know the smooth ER. Rough ER is all that you're required to know about. And the distinguishing feature between that, again, is that the presence of ribosomes on the rough ER right here, and the smooth ER does not have does not have ribosomes. The Golgi apparatus, located right here in our diagram, um, products of the ER, so it brings in products of the ER and it modifies them to some degree. It stores them for a period of time, and then it usually moves them to other areas of the cell, or it will export them into the cell. And so here's actually, in our diagram, we have some molecules being brought into the cell. And actually, we'll talk about this process, but the plasma membrane kind of forms a bubble around them, and it pinches off so that we have a small vesicle. You can see that here. And that vesicle is going to move to the Golgi apparatus. The Golgi apparatus will modify them or kind of prepare them for the cell, and then eventually move that material to somewhere else in the cell where it's needed. The Golgi apparatus will also get rid of or release particles, and so we can see that. I guess there's not an image of that in our diagram here, but that's called exocytosis, and we'll talk about that. Ribosome, it's made of ribosomal RNA. It's actually made up of a specific type of RNA called rRNA, or ribosomal RNA, and as we've discussed a couple of times now, they are responsible for producing proteins. We'll look at that a little bit more when we get into transcription and translation. Mitochondria are extremely important in eukaryotic cells, and it's one of the distinguishing features between a prokaryotic and a eukaryotic cell. And they are the site of cellular respiration, and they use oxygen and sugars and fats to produce ATP. ATP is the form of energy, you could think of it almost as currency. It's the form of energy that the cell uses to carry out its functions. So if you eat something, or when you go and eat something, that eventually gets turned into ATP, which the cells use to drive the specific functions. We'll take a look at cellular respiration and photosynthesis in more detail towards the end of this first semester. So let's look at some of the differences, the things that distinguish prokaryotic and eukaryotic cells. Prokaryotes are single-celled. They do not have a nucleus or membrane-bound organelles. They're small, about 1 to 3 microns. A good example would include bacteria. The DNA is in a closed loop within the cytoplasm, and it has 70 S ribosomes. Eukaryotes are going to have a different type of ribosome, and the main difference between the two, ribosomes are made up of 2 different subunits. There's usually a smaller subunit and a larger subunit, and those 2 subunits come together in order to produce proteins. Well, prokaryotes have different subunits than eukaryotes. And so that's what this 70S ribosome indicates, is that the prokaryotes have a smaller, 2 smaller subunits than what eukaryotes have. Eukaryotes are usually multicellular. Sometimes they'll be single-celled, but usually they're multicelled. They have a nucleus and they have membrane-bound organelles. They're a little bit larger. They're quite a bit larger. They're about 10 to 100 microns. Some example would include everything but bacteria and archaea. So we humans, our eukaryotic cells, we are eukaryotes. Dogs, cats, plants, all eukaryotic organisms. The DNA is in a double helix, and it's contained within the nuclear envelope within the nucleus. Some things that they share in common, again, 4 things that all cells have: a cytoplasm, a ribosome, DNA, and a plasma membrane. And our last one here that got cut off, eukaryotes have an 80S ribosome. So the ribosomes for eukaryotes are, the subunits for the ribosomes are a little bit larger than what we would see in prokaryotes, and that's what this 70S and 80S is distinguishing, is the size and the subunits of the ribosomes. And that's based off of, the size is based off of how much rRNA makes up those different subunits of the ribosomes. So within eukaryotic cells, there's 2 different types: plant and animal cells. And for the most part, they're very similar, but there are some very specific distinguishing features. And so we'll take a look at those. The first off, you can probably notice just looking at this slide, is the shape or the size. Plant cells are usually more rectangular. They're often green because they contain chloroplasts, whereas animal cells are usually more of a circular shape, less of a rigid rectangular shape. So some of the differences between plants and animal cells, again, these are both types of eukaryotes. Differences between plants and animals: the plant usually has a large central vacuole. Oftentimes water can be stored there. And if we go back to our image, you can actually see here's our large central vacuole, and it takes up a huge proportion of the overall cell. Um, and so this is a very distinguishing feature, and you can actually see this very easily when you look at plant cells under the microscope. And as an easy way to identify plant cells, you'll see this large central vacuole within the cell. Animal cells will sometimes have vacuoles, but they're much smaller, and there's not a single large central vacuole. Plant cells can also sometimes have additional small vacuoles, but the large central vacuole is a very distinguishing characteristic. Plant cells also have chloroplasts. Obviously, hopefully you remember that these are used to produce or used in photosynthesis to produce sugars. We'll talk about photosynthesis again in more detail towards the end of the first semester, and we'll actually look at how that process works. But photosynthesis is specific to chloroplasts that are found in plant cells, not in animal cells. And lastly, plant cells have a cell wall to support their plant structure. It's on the outer end or the outermost portion of the cell, and so the cell wall is on the outside, and the plasma membrane, labeled the cell membrane here, is within or right next to the cell wall. And within plants, the cell wall helps to give it some support and some structure. Plants are growing generally very vertical. They grow pretty tall oftentimes, and so that cell wall helps to give it support and structure. A couple things for animals that are usually not found in plants. Animal cells have lysosomes. Sometimes they can be found in plant cells, but more often in animal cells. And as we previously discussed, animal cells have very small vacuoles. They do not have a large central vacuole like plant cells. Our last portion for looking at eukaryotic cells is looking at some of the extracellular components. And this means, or what extracellular means, is that we're looking at components of the cell that extend beyond the plasma or cell wall. The plasma membrane or the cell wall is the outermost layer of the cell, depending on the type of the cell, and sometimes we can have components of the cell that extend beyond that. And these components have very specific functions, and we're going to look at these in more detail in one of our upcoming sections. But basically, they are outside of the cell, and they help to serve the cell in some specific functions. Within animal cells, they will release proteins or have proteins on the exterior portion of the plasma membrane. So the blue in our image here is outside of the cell. This is within the cell. And outside of the cell, there are some proteins that help to provide support, adhesion, or movement. Oftentimes, these extracellular components are used to help recognize other cells or recognize chemical signals that are within the area so that cells can kind of communicate with one another. Within plant cells, the cell wall maintains cell shape, prevents extra water uptake, holds plants up against gravity, and so these extracellular components of plant cells are really helping it to kind of maintain shape in its structure. That's it for eukaryotic cells. In our next couple videos, we'll look at the cell membrane more specifically and see how the extracellular components help the cell to maintain its functions.